I'm very happy to introduce today Erin Mordecai to this EPPIcenter seminar. Erin is an ecologist. She first did her bachelor's degree in mathematical biology at the University of Georgia. She then obtained a PhD in UC Santa Barbara in ecology and evolution. And she now is an assistant professor in the biology department at Stanford, where she has her lab. And she does some really fantastic work on the ecology of infectious diseases, and in particular, or at least the work that I know of her, is on vector-borne diseases and the interactions between environment and vectors, and how that ultimately impacts transmission. And she combines both modeling and empirical work. And also, we like Erin's lab very much because they were the first to put together the Bay Area EID conference a couple of years ago, and that's still ongoing, and that's really a great initiative to bring the people from the Bay Area doing modeling of infectious diseases together. So yeah, we're very happy to have Erin today here.
Thank you, Isabel. Thanks so much for the invitation to be here. I'm excited to speak with you all. I hope we can all meet in person fairly soon because we're pretty close to each other. Let me get my slides up here. Okay, does that look good? All right, awesome.
Yeah, that looks great.
So yeah, today I'm excited to share with you some of the research we've been doing in my lab over the last 6 years on how global change affects the ecology of vector-borne diseases. But before we dive into that, I want to go back in history a little bit and talk more historically from this perspective, how vector-borne diseases have shaped the course of human history. This is the tsetse fly. It's a really biologically amazing organism that gestates just a single egg in her uterus for months at a time before laying a single larva, which then takes a month in the soil to develop and hatch out into a tsetse fly. So for those who are familiar with mosquitoes, this is a very different biology. It has a really restrictive range of temperatures and humidities that have constrained it to a particular region of sub-Saharan Africa that has a suitable climate. And this tsetse fly has never dispersed outside of sub-Saharan Africa. It transmits a group of trypanosome parasites that cause what's called human African trypanosomiasis or sleeping sickness in people, and nagana in animals, which can be a really severe animal disease. It has a really long coevolutionary history with all the wildlife in Africa. And so as a result, thank you. As a result of this long coevolutionary history, many of the native wildlife species in Africa are what's called trypanotolerant. So they've evolved the ability to not have severe disease from this trypanosome parasite. But many of the introduced livestock and other domestic animals get severe disease from this trypanosome transmitted by tsetse flies. So as a result, in much of sub-Saharan Africa, in what's called the tsetse belt, we never had the development of the plow, basically. We could never have the plow because you never had domestic animals that could pull a plow because the tsetse fly was transmitting this really devastating parasite. And this has shaped the whole pre-colonial and post-colonial development of Africa because of the systems that built up around this more kind of low density distributed societies that didn't have a lot of centralized organization. And one of the really interesting exceptions that supports this rule is the Great Zimbabwe Society, which formed between about 1000 and 1400 years AD in Zimbabwe, in this place that's been described as a peninsula in a sea of tsetse. So it's a high elevation plateau between the Zambezi and Limpopo rivers, where this civilization built up a huge density, about 18,000 people, with this amazing architecture, these granite blocks that were hewn from the stone and converted into this amazing architectural setup. That includes, these walls that you can still see today, as well as intricate drainage that still works today. And this society traded with other societies as far-flung as China and Syria. And all of this was possible because there was no tsetse fly climatically in this region. So then zooming forward a few hundred years in time and to a different part of the world, the transatlantic slave trade introduced Aedes aegypti mosquitoes, yellow fever virus, and malaria parasites to what's called the New World, the Caribbean, Latin America, North and South America. And over the 200 years or so of colonial history leading up to the American Revolution, these parasites became really well established in much of the Americas and the Caribbean, to the point that during the American Revolutionary War, continental soldiers who had grown up in parts of the South where malaria was endemic, like the Low Country of Carolina, Georgia, Florida, Louisiana, people had this repeated exposure to malaria, much like people in the rest of the world that are repeatedly exposed to malaria. You build up sort of a clinical resistance that prevents you from getting really severe disease after a lifetime of malaria infection. And because a lot of the continental soldiers had this resistance built up and the redcoat soldiers, the British soldiers, did not, when Cornwallis attempted to invade this Chesapeake region, his troops were completely decimated. About 50% of them were knocked out with malaria. And basically the rebel troops were able to win the battle and ultimately the war because this forced Cornwallis to surrender at Yorktown, which was a huge turning point in the American Revolution. So again, disease has this huge impact on human history, in this case through differential burdens because of differential previous exposure. Going forward in time a little bit more to antebellum New Orleans. This is a map or a drawing of what antebellum New Orleans looked like in the 1840s. During this period, yellow fever epidemics occurred really regularly and really devastatingly, killing tons of people. And it was a really feared, really severe disease that had been established in this region. So understandably, people who were immune to the disease, so if you have it in childhood and you survive, you have lifelong immunity. And so people who had this immunity were sort of viewed as sort of better targets for social investment. So people could receive jobs, they could receive loans, they were more eligible for marriage. And this concept of immunocapital, as coined by the historian Catherine Oliverius, took hold where people that had this immunity could sort of benefit from more investment, but this was applied really unequally. And in fact, only white men could have immunocapital. Women and Black men and women were not afforded this immunocapital. So it was applied in this really discriminatory way. And in fact, the biology of yellow fever immunity was twisted. The understanding of that biology was twisted in a racially stereotyped way to, people of the day, doctors of the day asserted that Black people were naturally immune to yellow fever and therefore morally obligated to be enslaved and to work the land. And so this totally twisted view of the biology of immunity to yellow fever, which is of course not true, was used as a justification for enslavement while also benefiting white people that were already in power. So these are just a few of the examples that we came across when we reviewed the literature on how vector-borne diseases have shaped the course of human history. We found examples from across all inhabited continents on Earth. So you can see the little map down here in the bottom corner. And a lot of the key examples came from some of the most prominent human vector-borne diseases like plague, malaria, yellow fever, and trypanosomiasis, as I mentioned in the beginning. And we can summarize a lot of these impacts through the idea of a socio-ecological cycle, a feedback cycle. So if we start here with the human-made environment, humans, of course, always modify our environments around us. And those modifications affect the ecology of the vector, which in turn affect the disease burden, which then leads to a societal response to that disease burden, which can sometimes feed back to influence the human-made environment. So some examples of this, of human-made environments, are agriculture, urbanization, deforestation. These, of course, affect the ecology of the vector, the amount of breeding habitat, its contact with reservoir hosts and pathogens, which in turn affects disease burden, both morbidity and mortality, and often in a differential way across populations that have different histories of exposure. And then these can lead to social consequences. They determine the impacts of colonization, immunocapital, social and racial hierarchies were developed around disease responses, and infrastructure, things like piped water and sanitation, were built in response to these disease risks. And so this framework of socio-ecological feedbacks of vector-borne disease on human history carries forward today. Of course, vector-borne diseases remain a really important burden on humanity. We see about 96 million cases of symptomatic dengue every year. That's an estimate. 2.6 million healthy life years are lost to schistosomiasis every year, and over 400,000 deaths occur due to malaria every year, despite all the efforts towards malaria elimination and eradication. So this remains a really big and important problem today in humanity. So the question that most of my research addresses is, how is global change today altering infectious disease dynamics, taking this kind of socio-environmental feedback approach? And in some ways, this is a challenging question to address because vector-borne diseases, and in fact, most infectious diseases, are embedded within complex ecological communities, which involve interactions among a number of different species. For many vector-borne diseases, there can be multiple vectors, multiple host species. Most parasites interact with other parasites, either within host individuals or within host populations. And then multiple aspects of the environment and environmental change affect both the host and vector communities and the parasite communities. So there's complexity here, clearly, and there's lots of opportunities for nonlinearity, which basically means a situation in which an incremental change in one of these variables doesn't lead to a simple, straightforward incremental response in the disease system, which makes these changes really hard to predict. So in response to these two challenges, most of the research in my lab has taken a data-driven modeling approach to try to incorporate these complex and nonlinear mechanisms so that we can better understand and predict how global change is affecting vector-borne disease. And the work I'm going to present today was done by a group of really amazing postdocs and grad students who have joined my lab over the last 6 years. So I want to take a moment to acknowledge all of their amazing thinking and hard work on these projects, as well as the NSF, NIH, and several of the Stanford institutes and centers that have funded our work, and a number of external collaborators who many of you may know, listed over here on the right side. And before we jump in even further, I'll just stop and define what I mean by vector-borne disease here. These are pathogens that are transmitted by biting arthropods like mosquitoes, fleas, flies, and ticks. And these are important because, of course, they cause a huge burden of disease in humans, diseases like malaria, yellow fever, Lyme disease, dengue. And they're also ecologically interesting because, of course, the vectors are these small-bodied ectotherms, which are living in and responding to the environment around them. So there's sort of a mechanism that links human impacts on the environment back to feeding back and influencing our health. And so today I'm going to talk about two kind of research programs in my lab. First, we'll talk about how climate change impacts vector-borne disease transmission. And then in the second part, which will be the shorter part, we'll talk about the impacts of land use change on vector-borne disease transmission. So how will climate change affect vector-borne disease transmission? First of all, I want to thank Crane for this picture here, this awesome picture of a mosquito on a thermometer. This is really motivated by this question: is a warmer world going to be a sicker world? This is kind of a paradigm that emerged around 2000 or so in the infectious disease literature, and in particular in the malaria literature, suggesting that, we know malaria is a tropical disease. We know that its transmission can speed up with warmer temperatures. So therefore, warming temperature might mean a lot more malaria transmission in a future world. And this is just one example of that type of model that projected that a warmer world would be a sicker world. This is showing sub-Saharan Africa and the change in the number of infectious mosquito bites per person per year, showing a projected increase of up to 400 additional bites or more per year by 2050 because of climate change. But what our research wanted to address is it possible that the responses to climate change will be more nuanced than simply warmer is sicker in a lot of different systems? And we're thinking about that because if you think about the life cycle of a vector, like a mosquito, it's really complex, and there's lots of different ways that temperature can influence that vector life cycle. So the mosquito, the adult female, has to survive long enough to lay some eggs on the side of a body of water. Those eggs then have to hatch and go through 4 larval stages of development, pupate, and then emerge as an adult. Each of those depends on the metabolism of the mosquito, which, since the mosquito is an ectotherm, its metabolism depends directly on temperature. And then in order to lay those eggs, the female mosquito has to bite a vertebrate host and take in a blood meal, which she then converts into egg production. And her rate of biting also depends on her metabolism, which depends on temperature. And then the parasite, for its part, has hijacked this mosquito life cycle to transmit pathogens between one host and another. And as many of you know, this is not just like a flying syringe that takes parasites from one person and puts them directly into another person. There's this exquisite biological process in which the parasite, rather than getting digested and excreted, it is able to break through the midgut barrier of the mosquito, disseminate throughout the body, and then bind to the salivary glands, at which point the next time that mosquito bites, she'll inject parasites into that next host. And that's called the extrinsic incubation cycle. So the rate at which she completes that cycle and the probability that the vector actually becomes infectious are both dependent on the mosquito metabolism and therefore on temperature. So if we look across this whole life cycle, we can see lots of complexity here. We can see lots of different ways that temperature can influence the mosquito transmission cycle. And more generally, when we think about the impact of temperature on these different traits of the mosquito and parasite, we have strong expectations from ectotherm physiology about how those traits should respond to temperature. In particular, we expect that at some minimal temperature, the critical thermal minimum, the performance of a trait like development or survival or biting rate goes to 0. And then above that temperature, the trait can increase up to a thermal optimum and then decrease often fairly steeply up to a critical thermal maximum, where again, the trait goes to 0. And this is one of the most well-supported patterns in all of ecology. It's supported by empirical data across a wide range of different ectotherm taxa and traits. And it's also really well supported by theory based on first principles of enzyme kinetics. And yet, despite this strong expectation for nonlinear or unimodal responses to temperature, we often see vector-borne disease models making the prediction that temperature will linearly increase trait performance, which is what has led to this warmer is sicker paradigm. So the goal of our research is to reconcile this disconnect between nonlinear thermal performance curves observed in other ectotherm taxa and what's often assumed in these vector-borne disease transmission models. So how does temperature affect vector-borne disease transmission? That's our specific question here. We had a few specific hypotheses we wanted to test. The first is that both the traits of the mosquito and parasite and the transmission process as a whole respond non-linearly to temperature. We hypothesized that thermal responses would differ across different vector and parasite species, and that thermal responses will predict the spatial distributions of disease. But when we're trying to predict the temporal dynamics of disease, we need to know not just the climate, but also the susceptibility of host populations. So we tested these hypotheses first by developing empirically derived thermal performance models for each of the traits that's relevant for transmission. So we went into the literature and we found experiments where people had measured biting rate, mortality rate, demographic rates related to mosquito abundance, the transmission and infection probability, and the parasite development rate across different constant temperatures. And we took those empirical data and we fit a continuous thermal performance curve to each of those traits. So then we have an estimate of how temperature affects each trait individually. We can then synthesize that to get an overall relationship between temperature and transmission using a model called R0, which I'm sure many of you are familiar with. This is the number of secondary cases caused by a single infectious case within a fully susceptible population. And for our purposes, R0 provides sort of a static snapshot of how suitable a given temperature is for transmission, with all else being equal. So we can calculate R0 using a slight modification of this function that's been used and developed over 120 years. And the important thing to notice about this function is that it's a nonlinear function of many of these mosquito and parasite traits, which are themselves nonlinear functions of temperature. So this is where the complexity and the nonlinearity can really get integrated into our estimates of how temperature as a whole affects transmission process. And then we can take these empirically derived estimates of R0 from the lab and test them against field observations to validate the models and make sure that we're really describing the transmission process accurately with respect to temperature. And then we can take those models and project them forward to try to understand current and future transmission risk under changing climates. So we applied this consistent approach across 11 different mosquito-borne diseases, starting with Falciparum malaria, which was the work that Crane was involved in, and then working on dengue, chikungunya, Zika, West Nile virus, and a number of other important human vector-borne diseases. And what's important is we did this in a consistent methodology so that we could then compare and contrast thermal responses across these different systems. So synthesizing this body of work that we've done over the last 10 or 12 years or so, we find really consistent support for that hypothesis that trait thermal responses are nonlinear. So this is just one example of thermal performance data for mosquito development rate from Culex and Eulorostris, a mosquito in Australia. But you can see consistently that there's nonlinear thermal responses across this whole range of different mosquito species that we looked at, including the major vectors of dengue and chikungunya and Zika, these Aedes vectors, these Culex vectors of West Nile, and these Anopheles vectors of malaria. So we can now compare and contrast those thermal response functions across different traits and across different taxa. So each of these panels shows a different mosquito trait and its response to temperature, and each line shows the empirically measured thermal performance curve for each of these different mosquito species. And what you can see just glancing at these plots is both some consistency in the shape of the thermal performance curves. They tend to be nonlinear and most of them are hump-shaped, but also some differences among different species in how exactly they respond to temperature. And we see that same pattern when we look at the traits that involve the parasites as well. So things like transmission and infection probability, vector competence, and extrinsic incubation rate. So we have these 88 different empirically derived thermal performance curves, and we can now take the thermal performance curves for each pairing of vector and pathogen and calculate an overall temperature-dependent R0 estimate for those systems. And what we find here is consistent support across these systems for a unimodal or nonlinear relationship between temperature and transmission, where we have some consistency in the shape, and yet we have some variation among systems in exactly where their thermal optima are, where their lower thermal limits are, and to a lesser degree where their upper thermal limits are. So if we zero in on a few specific pathogens of interest, for dengue and Zika transmission by Aedes aegypti, we predict an optimum of 29 degrees Celsius. For Ross River virus, which is an important arbovirus in Australia, we see a thermal optimum around 26. For Falciparum malaria, that thermal optimum is around 25 degrees Celsius. For West Nile, it's about 24, 23, depending on the vector species. And for Eastern equine encephalitis, which is really common on the East Coast, we see one of the lowest thermal optima of around 23 degrees Celsius. So now we have this kind of predicted relationship between temperature and transmission across these different pathogens. But how do we know that this is actually relevant to what happens in the field? And this is arguably, this validation step is one of the most challenging parts of this research, because as you probably are aware, working in infectious disease, there's always data availability and bias issues. It's really hard to get direct measures of transmission in the field across a range of temperatures where only temperature is varying and not other things. So we have issues like spatial aggregation, variation over time lags, and multiple concurrent drivers going on. So as a result, we haven't been able to really do, in many cases, a statistical analysis, but instead we've simply plotted the observed incidence or transmission rate against temperature and compared that with our independent empirical predictions. So for Falciparum malaria, this is our temperature-dependent R0 estimate, which peaks around 25 degrees Celsius. For comparison, when I mentioned those warmer is sicker models that use the linear thermal responses, they predicted about a 7-degree warmer thermal optimum for malaria transmission. So this is a really important difference between 32 degrees Celsius and 25 degrees Celsius. This is in part why we felt it was so important to validate these models, because we wanted to know whether transmission in the field really did support this predicted much lower thermal optimum. And what we found, we plotted the entomological inoculation rate, or the number of infectious mosquito bites per person per year versus temperature, which was measured in locations across sub-Saharan Africa over a 30-year period. And what we see, first of all, is that these EIR points tend to be highest around that empirically predicted 25-degree optimal temperature, and they tend to decline steeply as we get above 28 or so. So supporting that idea that this is a nonlinear pattern, it's not just warmer is sicker. But we also can see that there's quite a bit of variation here in these data that's not explained by temperature, as we would expect, because this includes variation in population sizes and densities and behavior and control practices and humidity and so on. So to try to get at the variation that was explained by temperature, we bin these data points by temperature. So there's 10 data points per bin. And in the filled circles, we're showing the maximum within each temperature bin. This, we sort of think, represents the places where malaria is least controlled by other factors, and therefore potentially most sensitive to temperature. So if we focus on these filled circles, we can see that the height of the filled circles, the maximum rates of transmission, gets higher as we approach the predicted optimal temperature of 25 degrees Celsius, and importantly, that it declines as we exceed 25 degrees Celsius. So these independent field data are supporting the predictions of the laboratory-based model. Now, the cool thing that I've gotten—
Can I ask a quick question?
Yeah,
when you are looking at temperature, because temperature obviously changes daily and seasonally, what kind of reduced metrics are you using?
Yeah, that's an important question. So this was the average temperature during the transmission season. So there's a lot of like temperature variation being averaged out here that we're sort of looking very crudely at, you know, does the average temperature during the transmission season match up with what we would expect to be the optimal temperature for transmission? Yeah. And
that includes day and night. So the average of day and night across the entire season.
Yeah.
Okay.
Yeah. So yeah, that's a great point. So the next thing that we looked at, a little bit related to this, is like, does this entomological risk metric actually translate into human disease risk? And does human disease risk respond in the same nonlinear way? And to test that question, we worked with Dr. Desiree Lebeau at Stanford, who has a cohort study in Kenya, where they've worked with over 2,000 children measuring malaria incidence over time across 80 different sentinel households. So what we're plotting here is the incidence of malaria as measured by smear positivity in the triangles versus the average monthly temperature during the month when that was measured. And what we see here again is support for this nonlinear relationship where the highest levels of incidence are occurring around that 25-degree optimal predicted temperature. And importantly, we see support for that declining transmission or that declining incidence occurring above 25 degrees Celsius. So again, there is pretty strong empirical support for this nonlinear relationship with an optimal temperature at 25 degrees Celsius. Now, the next thing we were able to do that was kind of cool is by thinking about the nonlinear relationship between temperature and dengue transmission, it can really help us to reconcile some of the previously contradictory or context-dependent findings that we found in the literature. So postdoc Jamie Caldwell synthesized different empirical studies from the field where people had measured effects of temperature on dengue transmission, and they had found a wide range of different effect sizes and even directions. So in some cases, temperature had strong negative effects on dengue. In other cases, temperature had strong positive effects on dengue incidence, and in other cases, there was no real effect of temperature. But if we rearrange these effect sizes based on the temperature at which the study was conducted, we can see that that helps us to understand why we see positive effects in cases where the temperatures are marginally too cool, negative effects in places where the temperatures are marginally too warm, and then really no effective temperature when we're in this kind of sweet spot range of about 25 to 32 degrees Celsius. So this helps us to reconcile this nonlinear expectation, helps us to reconcile some of the previous contradictions in the literature between these different places or context dependence. So finally, what does this tell us about both current and future climate suitability for these different diseases? So how will climate change affect the spread of vector-borne diseases? Clearly, there's a range of temperatures if you're starting out between, say, 12 and 24 degrees Celsius, where any increase in temperature is likely to promote transmission of all of these diseases, maybe to varying degrees. And if you're starting at 29 degrees or above, any further increase in temperature is only going to decrease transmission because you're going up and over the thermal optimum for all of these different systems. But what's interesting is we have this range of temperatures in the middle between about 25 and 29 degrees Celsius, where you really see different patterns between different diseases. Some of them are really rapidly increasing, others are rapidly decreasing, and others are going up and over their thermal optima in this temperature range. That really is the range of temperatures that a lot of places experience during their summer months. So zooming in on malaria and dengue is two of the most important diseases to look at here. We mapped the future temperature suitability for transmission of malaria across sub-Saharan Africa. So this is an overlay of the number of person months per year that are at highly suitable temperatures. So the upper 50th percentile of that temperature dependent R0 curve. And what we see here is that under the current climate, the hotspot of suitability multiplied by people is occurring in kind of coastal West Africa and Central Africa. But with unmitigated climate change, we expect to see that shifting towards the higher elevation Albertine Rift region of East Africa, where it's currently a little too cool for optimal transmission, but it's really densely populated. But at the same time, we're predicting that temperatures are going to become unsuitable for malaria transmission, or at least less suitable in much of these regions where malaria transmission is currently really endemic. Now, in contrast to that, if we look at the temperature suitability for dengue transmission by Aedes aegypti in Africa, again, this peaks at 29 degrees Celsius. So this temperature-driven hotspot is predicted to shift from the same region of coastal West Africa to broadly expand across most of sub-Saharan Africa, including both West Africa and these higher elevation East African regions that are also becoming more suitable for malaria transmission. Now, on the surface, it might seem like dengue suitability is an irrelevant question for sub-Saharan Africa because we don't see that much dengue in sub-Saharan Africa. But in fact, studies that have started to look for Aedes aegypti mosquitoes and test people, do seroprevalence studies for dengue and other arboviruses, have found pretty substantial prevalences of 30% in adult populations, for example, for dengue seroprevalence in a study in Kenya, suggesting that the virus and the mosquito vector are already there. And combined with urbanization, this could drive a shift away from malaria and towards dengue transmission in some regions. And this importantly could require a retooling of public health efforts, because the way you control these mosquito vectors is very different because of their differing ecology. And of course, the drugs and diagnostics that are useful for malaria are not useful for dengue. So it's an important, potentially emerging public health problem to consider in this region, along with the need to continue malaria elimination efforts. So the next question we wanted to ask is, do these climate-driven models actually predict vector-borne disease dynamics over time? So do the dynamics of weather and climate actually tell us anything about dynamics of disease? And to address this question, we worked in 2 different locations, well, actually 8 different locations with collaborators who were doing studies on dengue, chikungunya, Zika, and Aedes aegypti in Ecuador and in Kenya. So 2 really distinct, culturally and ecologically distinct settings where dengue, chikungunya, and Zika were being measured. And we developed these dynamic versions of our models where we use the thermal performance curves that I showed you earlier in an SEIR framework to project dynamics of dengue, chikungunya, and Zika over time as a function of the observed weather in these locations. And what we found is that these arboviruses are highly predictable using mechanistic models. So here I'm showing observed on the x-axis versus predicted on the y-axis, and this is the number of epidemics here on the left, the timing of those epidemics in the middle, and the duration of those epidemics on the right. And we can see that across both the sites in Kenya, which are shown in triangles, and the sites in Ecuador, which are shown in circles, we see pretty high predictability of these features of when and where epidemics occur. By contrast, with our mechanistic models, we were not able to effectively predict the outbreak size very well. And we hypothesized that either the total size of the outbreak or the maximum number of infections that occurred during the outbreak. And we hypothesized that that's because host susceptibility modulates the effects of climate on disease dynamics. And so finally, to test this hypothesis that susceptibility modulates climate effects, we turn to sort of an emerging paradigm in ecological modeling called empirical dynamic modeling. The way this works, this has been pioneered by George Sugihara and colleagues, is if you have time series data of multiple variables that are potentially causally linked to each other, you can plot them in phase space. So what was the value of temperature, rainfall, and dengue incidence at any given point in time? And we can kind of trace through that phase space over time to reconstruct what's called a dynamical attractor. And then using the features of this dynamical attractor, we can understand both the causal relationships between these variables by looking at how much information about one variable is contained in the time series of another variable. And we can also do forward prediction based on sort of moving through this attractor and using the attractor directly to make predictions or forecasting. So we were able to apply empirical dynamic modeling to dengue transmission in San Juan, Puerto Rico, because of this dengue forecasting challenge the CDC put on a few years ago, where they gave this amazing weekly dengue incidence time series over a 20-year period, matched with temperature, rainfall data. And we were able to mathematically infer from the incidence of dengue, this index of host susceptibility. So we used these 4 time series to reconstruct our dynamical attractor. And first we asked, are these dengue dynamics predictable using this empirical dynamic modeling approach? And we found that yes, in fact, if we have information about climate and host susceptibility, we can do a pretty good job of forecasting dynamics of disease. So the true dynamics are in black and the predicted dynamics are in teal. We had a correlation of about 0.75 between those two in this 20-year or so in sample period, as well as this 4-year additional out of sample period. We had high predictability. But more importantly, what we wanted to know here is not just can we predict dengue dynamics, but can we use this modeling approach to understand the drivers of dengue dynamics and their interrelationships. So turning back to our mechanistic model of temperature-dependent R0, if we look at the slope of the effective temperature on incidence at any given point, we would expect that slope to be highest at around 25 degrees Celsius and to be less steep but still positive at these lower and higher temperatures. But we would also expect to only see these effects of temperature when host susceptibility is high enough to allow an epidemic to occur in that season. And so we divided our time series into settings where the inferred index of susceptibility was low versus high, and we plotted the slope of temperature versus dengue incidence. And what we see here is that when the host susceptibility index was low, these slopes were mostly near zero because there was basically no way for temperature to influence dengue if there weren't enough susceptible hosts. But when the host susceptibility gets high enough, we then start to see large positive slopes around that predicted 25 degree kind of maximum acceleration period. And we start to see those slopes declining back towards zero and even becoming negative as we approach the thermal optimum. So to summarize the effects of temperature on mosquito-borne disease transmission, we found support for the hypothesis that the trait responses to temperature are nonlinear, and that the thermal optima and limits of these trait responses vary across different taxa and different traits. We projected that climate change would shift the suitability for transmission, both geographically and seasonally. And then within a given region, you might see shifting climate suitability for different vectors and different pathogens. And finally, we found that climate and host susceptibility combine to drive disease dynamics over time. And an important caveat of all of this is that it's going to be mediated by human activities. So it's not just the climate that drives these things alone. It's also how humans respond to the changing climate and to the changing disease risk. And that sort of leads us into the second part, which is how will land use change affect vector-borne disease? This is one of the major impacts that humans are having on vector-borne disease systems in general. So this is work done by Andy McDonald when he was a postdoc in my lab, asking how does deforestation affect malaria transmission in the Amazon? And if you look at an aerial image of deforestation in the Amazon, contrary to what you might have expected, contrary to what I expected, you often see this kind of fishbone pattern. You don't just see clear cutting of huge swaths of the Amazon all at once. You see this pattern where a road or a river provides access into the forest, and then you get really intensive deforestation right around that river or road and the offshoots coming off of that. So you get this almost fractal-like pattern of deforestation where you get intensive deforestation occurring right along the edges of intact forests where people are living and working. So this type of deforestation tends to maximize the amount of forest edge habitat. Malaria in this Amazon region is transmitted by Anopheles darlingi, which is a forest edge associated mosquito. It breeds in pools of water that can accumulate during disturbances, and it transmits primarily Plasmodium vivax and Falciparum in this region. And what we've seen in Brazil over the last few decades is this really rapid resurgence of malaria, starting from the low point in the 1960s. And then in the 70s, we started to see this rapid upswing in malaria, both total malaria in black, as well as the individual parasites, vivax and falciparum, in red and blue. And this coincides in time somewhat closely with a period when the Brazilian government was actively promoting development in the Amazon. So the Trans-Amazonian Highway was built. Tracts of land were being given out for subsistence farmers to go and clear the land and live and work there. And following this period, we see this rapid resurgence of malaria in the Brazilian Amazon, providing a temporal suggestion, at least, that this could be responsible, this land use change and deforestation could be responsible for the increase in malaria transmission. If we look at the patterns over space, this is a snapshot from 2008 showing Brazil with the Amazonian municipalities and the colors. This is the Falciparum incidence in red. The vivax incidence spatial pattern looks very similar. On the right panel here, we have the forest cover map. What we can see is that most of the forest is already lost in this outer portion of the Amazon, whereas the forest mostly remains intact in this interior portion. And there's this sort of interface here between where the forest is mostly gone and where the forest is still there. And this is where we see our arc of deforestation, where the rates of deforestation are highest. So this is a year-on-year number of kilometers of forest loss. So looking at the spatial patterns here, we don't necessarily see a clear association. There might be some spatial correlation between deforestation and malaria, but it's not necessarily clear. And even if it were clear, that wouldn't necessarily tell us that it was a causal relationship. So evidence remains mixed on whether deforestation, or to what degree deforestation, promotes malaria in the Amazon. And a number of different case studies have been conducted in different regions, and they've found really mixed effects. So in some cases, you see that deforestation does promote malaria. In other cases, there's no real effect. In other cases, there's even sort of a negative effect where the places that have forest restoration and conservation are the places that have the most malaria. So it's far from clear what effect deforestation has on malaria in the Amazon region. So this was our central question: is deforestation in the Amazon increasing malaria incidence? And if so, what's the magnitude of this effect? And there were a few specific hypotheses we had here. So first, we hypothesized that deforestation increases malaria incidence by creating that forest edge habitat and bringing people in contact with the forest edge. But we also hypothesized that because malaria is a really severe and deadly disease, there could actually be a feedback of malaria on deforestation, where malaria risk could either directly or indirectly decrease deforestation, either by causing people to be too sick to work, or causing settlements to be abandoned, or otherwise being related to the economic processes that cause deforestation to decline. And so if there's this bidirectional relationship, that could be one reason that the effects of deforestation on malaria have not been clear so far. So we hypothesized this socio-ecological feedback between deforestation and malaria. The next hypothesis we had is that the relationship between deforestation and malaria varies across land use transitions, and that it would be strongest in these kind of natural ecosystem to frontier clearing stages, and less strong as we move through the more sort of intensified agriculture stages. So in the interior of the Amazon, where most of the forest is still intact, deforestation leads to this frontier sort of setting where you have frontier clearing, subsistence agriculture, small landholders who are living and working on their own land, sometimes in fairly unimproved housing conditions. Whereas in the outer regions of the Amazon, further deforestation only takes us closer to sort of intensified agriculture in urban areas. And there's larger landholders with forest edges that are further away. So we hypothesized that this deforestation and malaria relationship would be strongest in these frontier regions. So we wanted to address this question very broadly across the whole Amazon, and yet also ask a causal question of how does forest loss affect malaria? So to try to get at this using observational data, we borrowed some tools from the Econometrics toolbox, which is designed around understanding causal relationships in complex systems. So first, we used 13 years of malaria incidence data from 795 municipalities. We matched those up with remotely sensed forest loss and forest cover data. But we wanted to avoid any sort of spurious correlation that might sort of mistakenly lead us to conclude that there's a relationship when truly there's not. And so the first thing we did was include covariate data on things like climate, urbanization, and wealth that we would expect to drive malaria transmission. But then we can further control for variation that's unobserved by adding what's called fixed effects or dummy variables for municipalities and years. So this essentially compares a municipality to itself in years that differ in forest loss and malaria, and likewise controls for sort of year effects that happen across the whole region, such as an El Nino year or a shock in the stock market or something like that. So with these municipality and year fixed effects, we're dramatically reducing the chance of a spurious relationship driving our observed relationship between forest loss and malaria. And then finally, given that we have this hypothesis of a bidirectional feedback, we can statistically test for evidence of that feedback by fitting this whole model and then asking whether our error term remains correlated with malaria. And if so, that's evidence for endogeneity that could suggest a bidirectional feedback. To overcome that kind of endogeneity, we can use what's called an instrumental variable, which essentially the goal of this instrumental variable is to find something that's pretty strongly related to deforestation, but not otherwise related to malaria, except through its relationship with deforestation, and removes this potential feedback. So more concretely, what we used here was aerosol pollution, which is related to deforestation, but we don't otherwise expect aerosol pollution to have a relationship with malaria, except through its association with deforestation. And malaria in a given year is not expected to be able to feed back to affect aerosol pollution. So this instrument gives us an opportunity to see the causal effects in one direction of deforestation on malaria while eliminating the potential feedback. Then we can do this whole thing again, but reverse it. So now we're asking whether malaria affects deforestation. And in this case, for our instrumental variable for malaria, we're using the temperature suitability for transmission, which we calculated as the number of days that were between 24 and 26 degrees Celsius based on the mechanistic model that I showed in the first part during the transmission season. So it's a pretty narrow range of temperatures that we expect to strongly affect malaria transmission, but not otherwise independently affect rates of deforestation. So given all this buildup of how our methods work, the results are actually fairly simple and easy to interpret. In the first regression, we found that there's a strong positive effect of forest loss on malaria incidence. So here we're showing the Falciparum malaria incidence. This is our full fixed effects model with the instrumental variable for forest loss. And using our statistical tests, we did see significant evidence for that feedback, which did motivate our use of the instrumental variable. And once we include the instrumental variable, that endogeneity goes away. And more importantly, we estimate that the magnitude of the effect is fairly moderate. So about 6.4 additional malaria cases for each square kilometer of forest cleared. Now we can reverse the regression, and we can see evidence that, as hypothesized, Falciparum malaria incidence reduces forest loss. So again, this is our fixed effects model with an instrumental variable for malaria. Again, we saw significant evidence for that feedback, which motivated the use of the instrumental variable. And here our effect size is moderate, so about 0.07 fewer square kilometers are cleared for every malaria case. So suggesting that there is this detectable and measurable and fairly robust effect of malaria cases on reducing deforestation. Finally, we tested our hypothesis that this relationship varies across land use transitions by subsetting the data to just first look at the interior region, where we're mostly in this frontier subsistence agriculture stage. And here we see that our effects of forest loss on malaria are about twice as strong as they are in the data set as a whole. And likewise, we see a stronger negative effect of malaria on forest loss. So that bidirectional coupling between forest loss and malaria are strongest in this frontier region where people are living and working very close to the forest edge and without sort of extra labor that can come and fill in when someone's sick with malaria. By contrast, when we look at the outer regions of the Amazon, where most of the forest is already gone and we're in this intensified agriculture stage, we now see very little relationship between forest loss and malaria. So our slope is now much, much lower, and we see no evidence for a feedback in this region, as you would expect, because malaria is at very low levels anyway. So it's not expected to affect forest loss. So to summarize this part, we see evidence for a bidirectional feedback between deforestation and malaria. So within the forest, deforestation promotes malaria by creating that forest edge habitat for the vector and bringing people in contact with the forest edge. And deforestation declines with high malaria incidence. We don't exactly know the mechanism for this. We hypothesize that it could be that people are either too sick to productively work or they're abandoning settlements, but it could also be that there's some broader association with social and economic forces that we're not detecting in the model. And this is a key direction for future research for us, is to try to understand how malaria incidence could affect deforestation. And finally, we find that these relationships are strongest in the frontier regions of the Amazon, where we're at the earlier stages of deforestation. And to put sort of a current event note on this story, under Bolsonaro, the environmental policies have been really relaxed so that deforestation is happening at a much higher rate in places like the Jamanchim National Forest, which you can see here in this Google image picture. And about 10,000 additional square kilometers of forest were cleared, so about a 30% increase in deforestation between 2018 and 2019. Based on our model, we would project about 62,000 additional malaria cases due to that relaxation of forest policy. So what this type of model allows us to do is it allows us to put a direct human health cost on an environmental policy. So we can directly estimate how many new malaria cases do we get because of this change in forest policy, and hopefully incorporate this information into developing healthier forest policy going forward. And just to give a brief coda to this story, a student in the lab, Marissa Childs, did sort of a follow-up on this, looking at the impacts of small-scale gold mining on malaria in the Amazon. It's kind of similar ecologically in the sense that it creates this deforestation, this forest edge. It also creates these kind of open pits of standing water, and it places miners in very vulnerable conditions because they're living in really informal housing with low access to healthcare. And so Marissa hypothesized that small-scale gold mining would promote malaria transmission by very similar mechanisms to deforestation in general. So using some similar types of econometric approaches, she estimated the effects of gold mining on malaria. And she found that for each kilogram of gold extracted, you see about 0.5 additional Falciparum malaria cases and about 1 to 2 additional Vivax cases on average. And these dots represent different responses in different municipalities. So you can see that the effects can get as large as 50 additional malaria cases per kilogram of gold. But she then hypothesized that, as we know with Vivax malaria, there's a stage that can persist in the liver and periodically relapse. So that infection with Vivax malaria isn't necessarily related to where you originally got infected. You can get relapsing malaria in a new region far, well past when you initially got exposed. So her hypothesis was that for Vivax malaria specifically, we would see not only local impacts of gold mining on malaria, but also spillover effects where the surrounding regions would get malaria cases due to gold mining. So she fit a model with spatially lagged effects to test this hypothesis. And what she found is, as you would expect for Falciparum malaria, there's no long-term relapse. So we didn't see any significant effects of distance. But for vivax malaria, we see significant effects of distance up to 250 kilometers away. And a kilogram of gold extraction can cause about 50 additional malaria cases up to 250 kilometers away, resulting in a total impact that's much larger than just the local impact, suggesting that this is a really important process to consider when we're trying to think about malaria elimination in this region, because people are getting infected with malaria during gold mining activities in one region and then taking it with them and dispersing elsewhere. And if we do some calculations to try to estimate how much of the malaria burden could be attributed to gold mining, there's some uncertainty in these estimates, but up to about 24% of all vivax malaria cases could be attributable to gold mining in the Brazilian Amazon, suggesting that this is a really important pathway to consider and to try to come up with alternative, much more sustainable livelihoods to help resolve this environmental and health problem. So to conclude, how will global change affect the spread of vector-borne diseases? For climate change, we found that it's expected to have nonlinear and yet predictable effects on transmission. So it's not just that it's complicated and we can't predict, it's that based on the biology of the mosquito and the parasite, we can make pretty good predictions about how they respond to temperature. And we're likely to see shifts in transmission into different regions and different seasons based on the relationship between temperature and transmission by each vector. And one of our important future directions that we're excited about is measuring mosquito adaptation to temperature in the Aedes sierrensis, western treehole mosquito species system, which is a widespread mosquito species that occurs in naturally occurring tree holes from San Diego all the way up to British Columbia. So it experiences a huge range of climates in its natural habitat. So we're working on measuring thermal adaptation in that mosquito species. And then in the second part, we showed that land use change often affects infectious disease dynamics both through the vector ecology and the human behavior. And it's possible to have socio-ecological feedbacks where human behavior potentially responds to and impacts the vector ecology and vector-borne disease transmission. And one of our key future directions here is working with collaborators in both the Peruvian Amazon as well as in Costa Rica to try to understand the socio-ecological mechanisms as well as some potential solutions, ideally win-win solutions for the environment and for people's health in these different systems. And I'll just leave with this concluding message that I think environmental change has nonlinear and yet predictable effects on infectious disease dynamics. So thank you very much, and I'm happy to take any questions now.
Yeah, I have a comment about some of your initial thermal response work on malaria. Just curious if you've seen, because we have collected in our cohort studies in Uganda, we have collected lots of detailed data on entomological inoculation rates and incidence of malaria in three very distinct settings, but these are longitudinal studies over several years with highly detailed data on both incidence and EIR. So and it's all publicly available in, I mean, yeah, the full data set is published. I can send you the link. So yeah, we were wondering if you've seen it. Yeah.
Well, I have not.
Okay. Yeah.
I'm going to just write that down. We're actually working with Matt Thomas on it sounds like somewhat similar data set in the Ivory Coast, where they were developing this new intervention that they're testing. They had a randomized controlled trial, and they measured malaria incidence over 2 years, as well as a bunch of entomological data. But if you have a longer time series, that could be even more helpful. But what we're seeing even in that data set is there is a pretty detectable effect of temperature, even in a really highly endemic setting that has, you know, suitable temperatures all the time. Basically, it's not just driven by rainfall. There's a detectable effect of temperature too. Yeah,
I actually haven't explored in our data like the variation in temperature, right? Yeah. But yeah, it would be.
I would love to talk with you about that.
Yeah,
I'll get that from you. Thank you so much.
Yeah.
Isabel, just to add to that, and we can discuss more later, like right after or at another time, but there's also all the UMSP data, which is, you know, are more
for sure.
But it's 70 sites across Uganda. So,
but no EIR data, right? Like, no, I was thinking
no
about
EIR
this.
Data, correct.
But I agree. But yeah, I agree that that's also a source.
Like a validation set, for instance.
Yeah. Well, that's one really interesting question. Is like, if you have EIR data, how close does that get you to understanding human incidence? And like what we're finding so far in Matt's Cote d'Ivoire study is like, it's surprisingly not that helpful. Like,
but it's much more helpful than in dengue, for example. It's like
interesting. Interesting. I mean, in this particular study, what we found is like a lot of the information is just contained in climate, and like EIR is just this like messy thing that's sort of correlated to climate, like kind of disappointingly, but also maybe it tells you something useful that like, if what you're trying to do is to gauge human risk of malaria, maybe you don't need to do EIR, maybe you just need to do climate. But I'd be really interested in how well that compares in your Uganda study.
But yeah, there's a couple other things I was just going to quickly mention. You know, one of the things that's correlated with both incidence and with temperature is kind of seasonal nature of malaria, because we know at least at some level that later in the transmission season, people become kind of temporarily more clinically immune. So if you're looking at your outcome at incidence of clinical disease, that's going to drop off. And I assume that the temperature may also change over the course of a season. There are probably some gold standard data sets where you can look at the force of infection more directly. We're generating some of those, and there's a couple others out there where you're actually looking at the instance of new clones appearing in people's blood, but that are flying longitudinally. Those data sets will be more sparse, but that might be a way to at least explore some of these more complicated relationships between these broader ecological metrics and what's going on in a person.
Yeah, and that's exactly the same issue. And I think we've discussed this in the past with arboviruses, right? Like that incidence is dampened by immunity. So like trying to integrate the epidemiological data becomes, or validate against epidemiological data ultimately may throw you off, when in fact, what the reason why it's not fitting is more the immunity in the population.
Yeah, that's a great point. I mean, yeah, and I think there's like two related questions here. One is, you know, to what extent do our laboratory-based estimates of the optimal temperature actually bear out in the field? And in particular, the going above the optimum, does that actually slow down transmission? And then there's the question of just like, how useful is it to have climate information for predicting disease dynamics? Like how far can you get just by measuring the climate? Because that's pretty easy to measure. Or how much do you need these much more sophisticated things like EIR or human immunity data, which would be much harder.
But we'll never have those. I mean, we'll never have those at scale. So, right. I mean, I say never. I mean, who knows what we'll have in the future in terms of technologies. But climate data, we're always taking very sparse data sets, whatever model we're using, and projecting much more broadly in terms of risk of these diseases based on climatologic and other data.
And then there's other things that you can maybe use as proxies for these missing components of the data, right?
Yeah, I mean, and the point that I'd like to make is like people are using ever more sophisticated global general circulation models and different RCPs and different, lots of other like bells and whistles added on to these climate projections. But if you start with the assumption that traits respond linearly to temperature, it's always going to be wrong because the traits don't respond linearly to temperature. So it's like a lot of it's hard because a lot of these models are displayed as a map. So it's hard to see what's going on under the surface, and there's just so much complexity to it. Unless you really know what you're looking for, it's hard to know what the thermal response assumptions are. So I think the key point is like, there's lots of other stuff layered on top of these thermal responses, including like temperature variation, like Brian brought up, and immunity and lots of other things, maybe local adaptation, but fundamentally, like using a nonlinear thermal performance curve is much better than a linear one and is more likely to get us the right answer.
Yeah. I guess it's the constant tension between biologically motivated models as well and fully data-driven, let's say, machine learning-based approaches that essentially allow any kind of relationship or force, like without, but it's super, super interesting.
Thank you. I mean, I think that one of the things that can happen with like a statistical model or black box type model is that you get declines, you get these seasonal climate-driven declines that maybe you might infer are because of rainfall or drought or immunity, because those things co-vary with temperature. But like, if you're trying to extrapolate into future climate change, into climate scenarios we haven't observed yet, and those become decoupled or coupled in a different way than they are now, then it's important to know if like warm temperature per se can limit transmission versus, the correlation between warm temperature and rainfall regimes and other things.
Are there any other questions before, given that it's 2 already?
Can I ask a quick question? So that was very impressive, Erin. Have you ever in your models for Aedes vectors used the biting rate of 1 instead of making it temperature dependent, simply because Aedes is often known to bite every day, right? No matter if it already took a blood meal. Well, I'm just wondering if you ever,
That's a good question. So like Crane knows a lot more about what's under the hood of these models, but basically like, because it's, you know, instead of measuring the biting rate per se versus temperature in the lab, we've measured like the inverse of the oviposition cycle length and then assumed that that's proportional to the biting rate. But the point is it might not be, because they might just bite all the time because they like to bite. And so like, is the model sensitive to that? We did do some sensitivity analyses where we just like went through trait by trait and said, what if this trait were set to a constant? How much would that change the R naught model? And it doesn't have a big effect. Partly because I think biting rate has a similar thermal response to some of the other traits that are very, they're very left skewed. So they like the peak is very warm. And so biting rate is not what's limiting to transmission at those high temperatures or at the low temperatures really. But yeah, I mean, it's an interesting point that like, we don't usually have good lab estimates of how frequently mosquitoes actually bite. And you know, the longevity estimates too, people are always asking like, what can you really say about the true longevity of a mosquito versus temperature in the field versus in the lab? Like, don't they just all get eaten? And you know, longevity of a mosquito is one of the important drivers. Like, it's one of the key thermal limitations. But on the other hand, like if that were true, then I think we would just see a lot more transmission at warm temperatures than we actually do, like at extreme warm temperatures, because that's where the mosquito just isn't living long enough. Like even if it doesn't get eaten, it's only living a day or two, you know, in the field at these really warm temperatures. So there's no way for it to become infectious and bite someone else. But yeah, so the realism of lab versus field traits, I think, is always a big open question. Hopefully that's something that you're working on, Crane.
Not yet.
All right, should we thank Erin for a fantastic
talk.
incredible talk?
Yeah.
Thank you so much. And we'll do a smaller group chat for a few minutes, Erin, if you have the
That'd
time.
be great. Yeah.
And then
I just want to say thank you to all of you who came, and I've met some of you, but not all of you, but feel free to reach out if any of this was interesting and you want to talk more about it, or you're working on related things. I'd love to. And, you know, normally during a seminar visit, you get to meet a bunch of people. So I'd love to like meet all of you at some point, you know, and we can talk more. But yeah, so thanks everyone. Thanks for coming.